Okay, we are back. So, the last thing we need to cover on this slide that I jumped over the first time because I don't know, my brain's not working very well, is that so we know that CD45 will dephosphorylate. CD45 is a phosphatase. CD45 is also one of the proteins that's going to be expressed more and in different forms when the cell becomes a memory T cell. So these memory T cells will have high amounts of CD45 sometimes, and so that makes them easier to activate. Because more CD45 means a greater chance that CD45 runs into CD4 and activates that LUC kinase domain, and you get all the rest of the signaling. So, yeah, you just make more copies, and they're all swirling around, and then it gives you a greater chance of interaction. So, CD45 dephosphorylates. It is a phosphatase. What is it that turns this off, though? Well, it's CSK. And so I think of this as like the dog whisperer going, yeah, CSK. It basically stops LUC from doing anything. So C-terminal Sarc kinase is the full name of this thing, CSK. And it comes in and puts the phosphate back on this tyrosine, which then causes LUC to basically bite its own tail again. And so now the kinase domain of LUC will be inhibited. And so that's just going to be roving around, and eventually it'll find its target. And so you're on a timer. Like, LUC only gets let out for a little while, and then it gets phosphorylated back up and locked away. And if it is no longer associating with CD4, which is associated with all the CD3 and the T cell receptor, then there's nothing for it to signal. There's nothing for it to phosphorylate. So here's our little arrows. CD45 does this, and CSK takes it back to that. Activate, deactivate. All right, you got it. You got this. Okay, jumping ahead. All right, so all this is showing is that ITAMs are all around you. So you remember those FC receptor proteins? So here's FC gamma. They bind to IgG FC regions. Here's FC epsilon receptor, binds to IgE FC, like the bottom little handle part. These will have one or more chains. They're immunoglobulin-like, you better believe. And they have additional chains associated with them. So this says gamma, but it's not the same as the gamma part of CD3, for example. These will have ITAMs on them. And so it's just showing that FC receptors also have an ITAM containing protein. And so there's another kinase that's going to come along and activate this. For each one of these, there's a different kinase that's going to come close when they're interacting and activate it, or a different mechanism by which that's going to happen. But it's ITAMs all the way down, basically. That's the idea. So, this is just for interest, I would say. This is not central to our mechanisms. I want to really hammer the mechanisms because that's the stuff we're going to test over. Yeah. Don't tell anybody. Right. All right. So, what happens next? Well, you got your T cell receptor complex, your T cell receptor, your CD3 and friends. Let's see, gamma delta epsilon, and zeta chains sitting down there. And if we've already brought in CD4, which has already got LUC on it, and if CD45 has already activated LUC, then my goodness, LUC is going to phosphorylate all of these things it can get a hold of. And then once that happens, of course, ZAP70 is going to, boom, lock right on there, just like you see it doing. And then once ZAP70 is bound, now ZAP70 is ready, and it can go off and phosphorylate things. And so what's it phosphorylate? It phosphorylates Latin SLP76, among other things. Yeah, but these are the two that we need for our first pathway. And these are both basically scaffold proteins. We're going to build different complexes on these proteins. So these are useful little building blocks that can be joined up in a multitude of ways, basically. Lat, I remember because it's this purple thing and it looks long and tall. Yeah, honestly, yeah. Slip 76 is like a little slippery sausage. It looks like a little curved sausage to me. I don't know. Whatever. Remember it your own way. Yeah. Remember it your own way. So, what are some of the processes involved here? And we will cover these processes in detail, but overview first, details second. For example, there is one process that will lead to formation of phosphatidylinositol triphosphate. That's PIP3 right here, and we'll get into that. Phosphorylation of this from PIP2 to PIP3 makes it something that's pH domain combined. And the pH domain will interact only with PIP3 when it has all three of those phosphates on it. And so AKT is a kinase that has the ability to bind to an activated PIP3. So if the cell makes PIP3, it'll be a little platform. It like floats around in the membrane. It'll always be attached to the membrane, but it's going to move around the membrane. PIP3 is also a part of this complex. So there's our LAT, there's our SLP76, here's our PIP3. So there are like three different scaffolds here. Protein that holds a LAT to a SLP76 is called GADS. And you see that domain there? You see what that looks like? Little block, like a little doorway block like that. There we go. That is an SH2 containing domain. Yeah, of course it is. Yeah. So yeah, fine. And so it's going to bind this phosphorylated tyrosine that was phosphorylated by ZAP70. Yeah, fine. And once we've assembled this great big, fish hook of a complex, it is able to catch multiple fish. So for example, we can bind PLC gamma, which is a phospholipase, which is going to take one of the phosphates off of here and shut it down, or I don't know, do something like that. We'll get into that in a minute. We can bind VAV, and VAV's got a whole signaling cascade downstream of that. Or we could bind ADAP. Fine. These are just some of the wonderful things you can do when you've assembled the fishhook complex, if you like. We're not going to call it that because that's just how I remember it. It is the complex of LAT and SLP76 and GADS, one of each. Phosphorylated. Phosphorylated to the gills. All right. So, yeah. PLC gamma makes more inositol triphosphate. That's right. And diacylglycerol by breaking down other phospholipids. Okay, that's fine. We'll get to it though. We'll get there. Okay, so first pathway. Let's focus. So if you've got, let's go through the whole thing, CD45 activating, bumping into CD4, which is holding on to a LUC. CD45 activates the LUC by dephosphorylating it. LUC is now active and it phosphorylates all the little ITAMs. Following me so far? Yeah. The ITAMs are now a binding site for ZAP70, because of course they are. ZAP70 is now a kinase which can go off and phosphorylate LAT and SLP76. Neat. Once you've got phosphorylation on LAT, GADS will bind it because it's got an SH2 domain. It's looking for a phosphate to love. Yeah. And GADS will interact with SLP76. It just does. Yeah. Sue me. If a PIP3 wanders along next to this big fishhook-shaped complex, then we can actually attach some other proteins there. So we can bring in PLC gamma, and PLC gamma would be floating around inactive unless it can bind to a PIP3. And if you have a phosphatidylinositol triphosphate, PIP3, then PLC gamma will actually help you make some more of those. But it's still off because it needs a little help from ITK, is one of the protein kinases that can do this. And so ITK comes in riding another PIP3 molecule, and it's going to bind to a phosphate on SLP76, our little hot dog, and it's going to transfer a phosphate to, boop, the back of PLC gamma. And that's just like, tag, now you're it. And so with that phosphate and bound to a PIP3, we have now unlocked PLC gamma. And so now it can be the best phospholipase that it's able to be. So a phospholipase is a thing that breaks down phospholipids. Yeah, obviously, right? Okay. And so PLC gamma will do this. So this is inositol triphosphate, so PIP3. Phosphatidyl inositol triphosphate. Yeah, that's where I was getting mixed up. Phosphatidyl inositol triphosphate. So it's a membrane lipid on the bottom. It has an inositol, which is a 6-membered sugar. Here's a picture of an inositol up here. And there are 3 phosphates attached where 3 of these oxygens are on the inositol ring. The inositol is hooked up via a phosphate to the part of the lipid that's like going to be in the membrane, the hydrophobic part. So, yeah. So PLC gamma is going to cleave PIP3, it'll cleave PIP2 into diacylglycerol and IP3. And so PIP2 is going to be PIP3, but with 1 less phosphate. And so when that happens, what it's going to do is we've now made 2 separate signaling molecules. So we took an intact PIP3 like this. We have PIP2. So it's got 2 extra phosphates here, 1 phosphate joining, 2 hydrophobic tails at the bottom. There we go. We're getting this right. Hydrophobic tails, 1 phosphate to join, 2 phosphates on the side of the inositol. And what PIP3 does is it breaks it right there. So it's going to break it into the head group, the inositol, which now has 3 phosphates. And it's going to have the bottom part, which has 2 fatty acid chains. And it's a glycerol molecule that's holding them together. So it's called diacyl, acyl meaning fatty acid, glycerol. Glycerol with 2 fatty acid chains. Yeah. Fine. So there you have a molecule that is going to wander around in the membrane and go off and do signaling things, I believe. But inositol triphosphate is now going to be a molecule that can float around. And so if you've got PIP3 in the membrane, you can cut off the heads. Phospholipase C is basically a little beheading protein. And it's going to pop the head group off, and it can then float around, and it can bind to some other proteins. So first thing that'll happen is that we're going to the inositol triphosphate, so the head group, is going to bind to a calcium channel. And so this is in the cell. This is going to be in the ER, if I remember correctly. And the ER stores calcium. That's just one of its functions. So there's a lot of calcium inside the ER, not so much outside. You can use calcium binding dyes, and they'll just stain up the ER in a cell, and a lot of the cell is ER, so it looks pretty good. It looks pretty darn good. When that opens, that little gate, now there's a lot of calcium inside, not much outside, so passive diffusion, the calcium is going to equilibrate, which means that, you're going to bring calcium in here. And once calcium is released, we'll get to the next one, but some of this calcium will then activate other proteins, and so we'll get further signaling. So PLC gamma can break a PIP2 into diacylglycerol and IP3, and IP3 can dock onto a calcium channel, and it will deplete. It'll just dump all the calcium, not all the calcium, but a lot of the calcium out of the ER. And then the IP3 part is going to break down or fall off or something. And when that happens, then boom, we're going to snap that calcium channel shut. And then we're going to refill the calcium stores from the cytoplasm by re-importing the calcium. And so this ORAI1 is going to be the calcium importer for the ER. Yeah. So why would we want to dump a whole bunch of calcium out? Well, like these proteins have names like calmodulin and calcineurin. I think you can see where this is going, right? All right. So when that calcium channel is open, calcium flows into the cytoplasm, where normally there's not a lot of calcium. We store it all away like cookies in the cookie, jar. inside of the ER. So we keep all our calcium locked up. Yeah, yeah. This is the gate. And so when IP3 binds onto this on the inside, because it was cut off of a PIP2 by a PLC gamma that was activated by ITK because it was part of a complex with a PIP3 and SLP76 and GADS and LAT, which were phosphorylated by ZAP70, which was able to bind to CD3 because it was phosphorylated by LUC, which was able to be active because it was dephosphorylated by CD45. Yeah. What you want to do is go through this process. Go through it forward and go through it backwards as many times as you can, because every time you recite how the different things are connected, or write it down, or whatever it is that works for you, it gets a little stronger, and then you can remember it, and yeah, then you got yourself a working cascade. So I think we're going to do like 2 or 3 more slides, and then we're going to cut this section and move on to the next section, which is more signaling. Yeah, that's what you want, so that's what you get. Yeah. All right. So here are our players. We've got calcineurin, this little purple guy. We've got calmodulin, which would love to be holding on to 2 calciums, like a little crab. But there's no calcium around. Poor calmodulin. It will modulate its structure in response to calcium. That's probably how it got the name, right? NFAT over here is going to be a transcription factor. But right now it's all phosphorylated, and phosphorylated NFAT is switched off NFAT. Just because, yeah, with NFAT, when it is phosphorylated, it doesn't work. Yeah, that's it. So we got NFAT with these stupid phosphates on it, and it could totally go into the nucleus and totally start some transcription if only someone, some hero, would come along and take off the phosphates. Who could do that? Well, calcineurin. Calcineurin is a phosphatase, and there's not a special diagram for, like, not a special shape associated with phosphatases. There aren't that many of them in this book, so they just tell you it's a phosphatase, like CD45. Yeah. And so calcineurin can't work unless it's bound to a calmodulin. So you see this going? Yeah. The gate's open. Calcium whooshes out of the ER. Calcium can bind to the 2 little crab arms of calmodulin, which then modulates its shape. And now there's a landing spot for calcineurin, which is going to be a phosphatase that will only work when it's bound to calmodulin, which will only be possible if calmodulin is holding on to 2 phosphates, so it can't just be hugging itself like that, basically. Once that happens, then calcineurin will dephosphorylate NFAT, because of course it will. It's a phosphatase. That's what it does. And NFAT binds to calcineurin, and so it just goes snip, 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 come in for a little trim and a little barbershop, and now you're good. You're salon fresh, ready to take on the world. And once NFAT is dephosphorylated, it can then go right into the nucleus and act like a bookmark, be a transcription factor, and we will now get transcription of all the NFAT-driven transcription things that need to happen. So there you go. That's going to be one of the sort of T-cell activation things. We'll be sending NFAT into the nucleus via this long convoluted process. So go through all those steps. There are a lot of steps. Go through all the steps. Make sure you know the steps. And yeah, the next section has got all of its own language. And this is also stuff that will happen. These are sort of multiple pathways that lead to different levels and types of activation. And in any T cell, you may get all of them switched on. I think that's probably the most common thing when you get a real good, strong hit. But you can also have situations where one or more of them are switched on. Others are not very on. Some are highly on. And so it makes for a bit of complexity in the immune response. Yeah, that's why you don't always get what you want with the immune system. But you get pretty good and you get to live. So yeah, it's enough for me for now. All right, thanks. That's been part 2. Come back for part 3. I dare you.